So for over a century, most of us have walked around with this massive biological ego.
Oh, absolutely.
Right. Like we assume that when it comes to diseases, things like malaria or dengue fever, humans are the main character.
We think it's all about us.
Exactly. We get bitten, we get sick, so obviously we are the host.
Yeah.
But if you look at the actual biology and the sources we have today, we have it completely backward.
Yeah, biologically speaking, we are just the intermediate host.
Which is wild to think about.
It really is. The mosquito is actually the definitive host. That is where the parasite actually reproduces. We're just, the temporary waiting room for it.
So today, we're setting aside our biological egos to look at the actual main characters here.
The arthropod vectors.
Right, the arthropod vectors. We are doing a deep dive into a massive stack of microbiology and epidemiology notes. Our mission is to really unpack the intricate biological machinery of mosquitoes, ticks, and fleas.
And we aren't just talking about annoying bug bites here.
No, not at all. We're looking at how these tiny creatures act as highly specialized active partners in spreading, the world's deadliest diseases.
Yeah, and to put the scale of this into perspective for you, vector-borne diseases account for roughly 1/6 of the entire global infectious disease burden.
1/6? That is just staggering.
It's massive. We are talking about over 700,000 deaths a year. So decoding the biology of these tiny vectors isn't just some academic exercise. It is quite literally a matter of life and death on a global scale.
Okay, let's unpack this. Because to understand how they operate today, we really have to look at the sheer stubbornness of the medical establishment when this was first being figured out.
Oh, the pushback was incredible.
Because for the longest time, everyone was convinced disease just floated in the air, right?
Right, the miasma theory.
Miasma.
Yeah, until the late 19th century, the dominant belief was that diseases were caused by bad air. Emanations from rotting organic matter in swamps.
Which makes sense why they thought that, I guess, if you live near a swamp.
Sure, but it was so ingrained. The word malaria itself translates from Italian directly as bad air, malaria. Breaking that paradigm was incredibly difficult for scientists.
Even when the physical evidence started showing up. Like I'm looking at the notes on Patrick Manson in 1877. He was a Scottish physician working in China, and he actually proved that a Culex mosquito ingested the microscopic worms that cause lymphatic filariasis.
Right, he had the evidence right there.
But the establishment still fought the idea that insects were responsible for major human outbreaks. They couldn't accept it.
The resistance was fierce, yeah. Look at Carlos Finlay in 1881. He was a Cuban physician who proposed that yellow fever was transmitted by a specific mosquito, the one we now call Aedes aegypti.
What happened to him?
The medical establishment essentially laughed him out of the room for 2 solid decades.
2 decades? Just because the idea that a tiny bug could bring down a human was, too much for their biological ego to handle.
Exactly. It really took Ronald Ross winning a Nobel Prize in 1902.
Oh, for showing the malaria parasites developing in the gut wall of the Anopheles mosquitoes.
Right. That Nobel Prize really forced the issue. But the human cost to prove Finlay right before that was just staggering.
The notes on Walter Reed's Yellow Fever Commission in Cuba in 1900 are intense. Right. To finally prove the mosquito hypothesis, they used human volunteers.
Yeah.
And some of those people actually died.
They did. They allowed themselves to be bitten by infected mosquitoes just to settle the debate once and for all.
That's a level of bravery I can't even fathom.
It was a huge sacrifice. But it forced a total shift in public health. Instead of just trying to clean up environmental filth, they realized they had to target the insects directly.
Which changed everything.
It did. That realization is what enabled the targeted sanitary campaigns that finally allowed the construction of the Panama Canal.
Oh, right, because the French efforts had completely collapsed before that due to disease. But even then, the early 20th century view was still pretty primitive. The assumption was just that the bug was like a passive syringe.
Like a dirty flying needle.
Right. A dirty needle that pokes person A, gets infected blood on its mouth parts, and then flies over and pokes person B.
Which fundamentally misunderstands the science of what's actually happening.
Because it's way more complicated than that.
Way more. A housefly walking on garbage and then landing on your food, that is what we call a mechanical vector.
Okay.
It's just physically dragging bacteria on its legs. But a mosquito is a biological vector. The pathogen doesn't just hitch a ride, it completely relies on the insect's internal anatomy to survive and multiply.
So they aren't dirty needles at all. Based on the strict biological definition in these notes, they operate more like a dark room.
Oh, I like that analogy.
So the pathogen goes in as raw film, And it has to sit there, develop, and go through this whole internal biological process before the photo is actually ready to be handed to the next host.
That captures the required sequence really well, actually. To be a true biological vector, the arthropod has to acquire the pathogen develops or multiplies inside it, and then finally the arthropod delivers an infectious dose.
So the insect's biology is a mandatory part of the cycle.
Exactly, it's an active partner.
But if the pathogen has to physically mature inside the vector, that means there's a mandatory waiting period.
Right.
It isn't instantaneous.
Right.
Because I think one of the most common myths out there is that if a mosquito bites your friend who has a virus, and then it immediately flies over and bites you, you are instantly infected.
Yeah, people assume the contagion is immediate. But because of that biological development phase you mentioned, there is something called an extrinsic incubation period, or EIP.
Extrinsic incubation period.
Yes. When a female mosquito ingests an infected blood meal, she is not contagious right away. The pathogen has to migrate out of her gut, reproduce, and eventually make its way all the way up to her salivary glands.
So it's literally physically moving through her body tissues.
Yes.
That has to take time. So how long is this EIP for something like, say, malaria?
Well, for Plasmodium falciparum, which is the deadliest malaria parasite.
The one carried by the Anopheles gambiae mosquito.
Exactly. For that one, the EIP is roughly 10 to 14 days at typical tropical temperatures.
Wow, 10 to 14 days.
Yeah, the parasite has to undergo sexual reproduction in the mosquito's midgut, form a cyst, burst, and then send thousands of new infective forms to the salivary glands.
Wait, that means for almost 2 solid weeks after biting an infected person, that mosquito is flying around, potentially biting other people, but is completely harmless to them regarding malaria.
That's right. The pathogen is trapped inside the dark room, so to speak, still developing.
That's fascinating.
What's really fascinating here is the critical vulnerability in that cycle. Because a female Anopheles mosquito in the wild typically only lives for about 2 to 3 weeks total.
Oh, wow. That is a razor-thin margin.
Very thin.
The parasite takes 2 weeks to develop, and the mosquito only lives for 3 weeks. The pathogen barely makes it out in time.
It is a massive biological bottleneck, and public health actually leverages that exact bottleneck.
Really? How?
Well, when you hear about insecticide-treated bed nets or indoor residual spraying, the strategy relies entirely on that EIP timer.
Oh, I see.
You don't necessarily have to prevent every single bite to stop malaria. You just have to ensure the mosquito dies before that 10 to 14-day timer runs out.
Because if you shorten its lifespan by just a few days, the parasite dies with it before ever reaching the salivary glands.
Exactly.
That is such a brilliant way to attack the problem. You use their own biological clock against them.
It's very effective.
And we should probably clarify why the mosquito is taking the risk of biting us in the first place. Because they don't actually eat blood for food, do they?
No, they don't. This is a biological trait called anatogeny.
Anatogeny.
Right. Male mosquitoes never bite. They feed exclusively on plant nectar for energy, and female mosquitoes also drink nectar for energy, but they require the concentrated protein and iron found in blood to synthesize their eggs.
So they bite exclusively to reproduce.
Strictly for reproduction, yes.
And different mosquitoes have entirely different habits, which is also crazy to me. We've talked about the Anopheles mosquito, which prefers rural environments or like natural swamps. But the notes highlight Aedes aegypti, and their behavior paints a totally different picture.
Yeah, the ecology of the vector completely dictates where the disease spreads.
Right.
So Anopheles is a night biter that likes natural bodies of water. But Aedes aegypti, on the other hand, is highly adapted to human environments.
This is the one that transmits dengue, Zika, yellow fever.
And chikungunya, yeah. It actually prefers artificial containers, things like old tires, flower pots, or plastic buckets to lay its eggs, and it bites during the day.
So as human populations explode and we build these dense urban centers with plenty of artificial water storage, we've basically paved a superhighway for the Aedes mosquito.
We have constructed its absolute ideal habitat, yeah. That behavioral shift is a major reason why dengue fever has become a massive urban crisis globally. It just thrives in the densest cities on Earth.
Okay, so if mosquitoes rely on speed and this razor-thin biological timer to spread disease, ticks go in the exact opposite direction.
Oh, completely.
They play the long game.
Right.
And biologically, they're a completely different animal.
They are. Ticks are arachnids. They have 8 legs and no antenna, which makes them closer cousins to spiders or scorpions than to mosquitoes.
Gross, but okay.
And their life cycle is incredibly slow. A hard tick, like Ixostapularis.
It's the deer tick, right?
Correct. It typically only takes 3 blood meals in its entire life.
Just 3 meals? But wait, don't they live for a couple of years?
Usually a 2 to 3 year life cycle, yeah.
That's wild.
They hatch as a 6-legged larva, take a blood meal, drop off into the leaf litter, and then they spend months just digesting and molting into an 8-legged nymph.
Months.
Then the nymph takes 1 meal, molts into an adult, and the adult takes 1 final meal to reproduce.
Because they eat so infrequently, they have to be incredibly efficient at holding on to pathogens.
Very efficient.
I was looking at the notes on transstadial transmission, and it seems like once they acquire a pathogen as a larva, they just sort of pack it away and carry it with them through the whole molting process into the nymph and adult stages.
Yeah. The pathogen survives the tick's massive internal restructuring during those molts, but it actually goes a step further with transovarial transmission.
Okay, what is that?
For certain pathogens, an infected adult female tick can pass the infection directly into her eggs.
So the next generation hatch is already infected.
Exactly.
They are basically multi-generational sleeper agents.
That's a great way to put it.
Because of transstadial and transovarial transmission, they don't even need to bite an infected mouse or bird to pick up the disease. They literally wake up ready to transmit it on their very first bite.
And they are very, very patient about delivering that bite. There is this persistent myth that ticks wait in trees and just drop onto your head when you walk by.
Oh yeah, the old warning to look out for falling ticks in the woods. I grew up hearing that.
We all did. But ticks cannot jump, and they rarely climb higher than knee level. They employ a strategy called questing.
Questing.
Yeah. They climb up a blade of low grass, hold on with their back legs, and extend their front legs out into the air. And those front legs have specialized sensory organs.
Oh, wow.
They just wait there, sometimes for months, for a host to physically brush against the grass so they can grab onto the fur or clothing.
It's passive, but clearly highly effective. And what's crazy is it took us a surprisingly long time to figure out they were spreading disease right under our noses. I mean, the discovery of Lyme disease didn't even happen until 1975.
Right, in Lyme, Connecticut.
Hence the name.
Exactly. Doctors noticed a suspicious cluster of children diagnosed with juvenile rheumatoid arthritis. It took an epidemiologist named Alan Steer to trace the outbreak back to tick bites in the surrounding woods.
And then a few years later, a microbiologist, Wiedie Bergdorfer, isolated the actual spiral-shaped bacterium inside the Ixodes ticks.
Right, which is why the pathogen is now called Borrelia burgdorferi.
Okay, so mosquitoes use a ticking clock, and ticks use extreme patience. But fleas use a mechanism that is straight out of a horror movie.
It really is.
So if you're listening and you've ever had a flea bite, you might want to brace yourself for what's actually happening under your skin.
Yeah, this gets a little gross.
Very gross. The sources outline this biological hack discovered by Bacon and Martin in 1914 regarding how fleas spread Yersinia pestis.
Right, the bacterium responsible for bubonic plague, the Black Death.
Yeah, the disease that wiped out a massive chunk of Europe in the 14th century. The mechanism is called the blocked flea.
Yes.
And from what I'm reading, the plague bacteria doesn't just sit in the flea's stomach. It literally builds a physical wall.
It forms a dense biofilm. So when a flea ingests blood infected with plague bacteria, that bacteria multiplies and secretes this sticky matrix in a part of the flea's foregut called the proventriculus.
Which acts like a valve, right?
Exactly. And the biofilm grows until it completely blocks that valve.
So the bacteria intentionally starves the flea.
It does. The flea is physically incapable of swallowing new blood. It's starving.
And a starving animal panics. So the flea goes into a frenzy, biting everything in sight, just desperately trying to feed.
But because the throat is completely blocked, the new blood it tries to suck up hits that biofilm barrier and bounces back.
Oh, man.
And the mechanical force of that recoil flushes the plague bacteria directly out of the biofilm and regurgitates it straight into the fresh bite wound.
Throwing up plague bacteria into every wound, it weaponizes the flea's hunger. That perfectly explains the explosive spread during plague outbreaks.
It does.
Because if the fleas are normally feeding on rats and the plague wipes out all the rats, you are suddenly left with thousands of starving, panicked, blocked fleas with no food source.
And they become desperate. They seek out human hosts, spreading the bacteria with terrifying efficiency. It is a perfect, albeit horrifying, exploitation of the vector's anatomy.
So, knowing how devastating these vectors are and having the biological blueprints of how they operate, I think the obvious question for you listening is, why haven't we just eradicated them?
It's a fair question.
Right. We have pesticides. We have global health organizations.
And there was a massive attempt to do exactly that. Between 1955 and 1969, the World Health Organization led the global malaria eradication program.
Okay, so we tried.
We tried hard. The strategy relied on brute force, basically. Widespread, heavy indoor spraying of DDT to kill the mosquitoes, paired with chloroquine drugs to treat the parasite in humans.
And reading the notes, it worked in some places, like parts of Europe, the Caribbean, and Asia.
Yeah.
But it completely failed in sub-Saharan Africa.
Right.
The biology just fought back.
Well, the Anopheles gambia mosquito is simply too efficient a vector, and on top of that, the mosquitoes rapidly evolved resistance to the DDT, and the parasites developed resistance to the drugs.
Plus, you can't just carpet bomb an ecosystem with chemicals without massive cascading effects.
No, you really can't.
The sources bring up Rachel Carson's 1962 book, Silent Spring, which detailed how DDT was accumulating up the food chain. It was thinning the eggshells of birds of prey, just devastating local ecology.
And the pushback from that era led to a fundamental shift from eradication to what we call integrated vector management.
Which means what exactly?
It means the consensus moved toward breaking the transmission cycle using multiple targeted tools. Things like bed nets to exploit the extrinsic incubation period, targeted larval control, better drugs.
So managing them rather than trying to wage an unwinnable war against nature itself.
Exactly.
But managing vectors is still incredibly complicated, and the science isn't always neatly settled.
Oh, definitely not.
The notes actually highlight two fierce, ongoing debates that perfectly show how complicated the intersection of biology and public trust can get. And I want to be clear to you listening, we are just reporting on these neutrally, exactly as they appear in the sources, because they are heavily debated.
It's important to be objective here, yeah.
Right, so the first one is chronic Lyme disease.
Right. So the mainstream medical consensus, represented by bodies like the Infectious Diseases Society of America, is that a standard course of antibiotics clears the active Borrelia infection in most patients.
Okay.
For those who continue to suffer from severe fatigue or joint pain, the mainstream view is that they are experiencing a post-treatment inflammatory response.
So the immune system is still reacting, but the bacteria are technically gone.
According to that view, yes.
But there's a massive patient advocacy movement, along with some researchers, who argue that the bacteria actually survive the antibiotics.
Yes. They point out that Borrelia can form persister cells or hide in biofilms deep in the body's tissues, making it nearly impossible to detect with standard blood tests.
So they argue these patients have an ongoing active infection requiring long-term antibiotics.
Right, and it highlights genuine scientific uncertainty. The mechanism of how we verify a cure for a pathogen that can potentially burrow into deep tissue and evade detection is just incredibly difficult to nail down.
It really is. Now, the second major controversy outlined here is over GMO mosquitoes.
This is a big one.
Yeah, a company called Oxitec has engineered Aedes aegypti mosquitoes to carry a dominant lethal gene. And they've tested this in places like Brazil and Florida.
They have.
From what I understand in the notes, they release millions of these genetically modified males, which don't bite, to mate with wild females.
Right, and the mechanism relies on a self-limiting gene. When those modified males mate with wild females, the lethal gene is passed to the offspring.
What does the gene actually do?
It essentially overproduces a protein that interrupts the offspring's normal cellular function, causing the female larvae to die before they reach adulthood and can bite anyone.
I mean, it sounds like an incredibly precise way to crash a local population of disease-carrying mosquitoes.
It is very targeted.
But critics are deeply concerned about the unknown ecological variables, right? Like, if the genetic mechanism fails, or if a small percentage of female offspring survive and reproduce, you risk introducing new genetic material into the wild population.
That's a concept called gene flow.
Gene flow, okay. And there's debate over whether that could inadvertently create a more robust mosquito down the line.
Exactly. Both of these controversies really illustrate that when you intervene in a complex biological system, whether it is the human immune system or a local ecosystem, the ripples are hard to predict.
Very hard to protect.
And that is actually the core philosophy behind the One Health framework that major health organizations use today.
One Health. Because human health doesn't exist in a vacuum.
No, it is directly tied to animal and environmental health. You can't control West Nile virus by only looking at humans and mosquitoes. You have to look at the birds.
Right, the birds act as the reservoir host.
Exactly. The virus circulates in the bird population, and humans only get infected when a mosquito bites an infected bird and then bites a human.
So humans are often just incidental dead-end hosts for those pathogens.
Mostly, yeah. If you get West Nile, your viral load usually stays too low to infect the next mosquito that bites you.
Hmm.
And the environmental pillar of One Health is especially relevant right now due to climate change.
Oh, because as average temperatures rise and winters become milder, the geographic ranges of these vectors are expanding.
Exactly. We are seeing ticks and Aedes mosquitoes surviving and breeding much further north than they historically ever could.
The ecosystem is shifting.
It is shifting rapidly.
And that really brings us back to where we started, looking at the true main characters of this story. Mosquitoes, ticks, and fleas are not just random, annoying pests.
They are highly adapted biological incubators.
They use ticking clocks, generational transmission, and horrifying anatomical hacks to keep these pathogens alive. And with rapid urbanization and climate change, this intricate biology is increasingly playing out right in our own backyards.
The complexity of these transmission cycles is astounding. I mean, we talked earlier about how the plague bacterium manipulates the flea by blocking its gut to force it into a feeding frenzy to spread the disease.
Right, weaponizing its hunger.
Well, as climate change rapidly alters the habitats and the stress levels of these vectors, you have to wonder, how might pathogens be currently evolving new, subtle ways to manipulate vector behavior?
Oh, wow.
Perhaps they are actively changing a mosquito's attraction to human scent, or altering a tick's questing habits, all to ensure their own survival in a rapidly changing world.
That is a terrifying but absolutely fascinating thought. The biology never stops adapting. The invisible war just keeps evolving.
It really does.
So to you listening, keep questioning, keep exploring, and remember, the next time you swat away a bug, you might just be looking at the definitive host. Thanks for joining us on this deep dive.